that. So for the last few years, I've been working as a data man community manager, and I have been associated with IBISBA for the last 4 or 5 years now. So without wasting any more time, what I'm going to tell you today is that we're going to briefly discuss why we need a data management hub like the IBISBA Hub or the Fair Data Station. Why is data management important? And I'll talk you through the features of the IBISBA Hub. And then after that, I'll take you through a live demo which will familiarize you with the IBISBA Knowledge Hub. It might be a lot of information, but that is the reason why we are recording this session, so that you can have access to all this information and you can see it at your own in your own time. So what I wanted to start with is that why do we need to manage data? What is it that we will get out of it? So data is your most valuable asset from all the research that you do. And practically, this is what our science is built on. Whatever data we create, we analyze it, and then people from our communities, scientific communities, or outside industry, they use this and take the research further. So it's worth it to invest time and energy in its management and maintenance. Then we also need to be very sure of the quality of data that we are presenting or creating, and that because it is essential for reliable results. Now, once we have done that, because we are a community and because our new research of science or new science is based on the previous research that has been done by our collaborators and colleagues, so our data should be shareable and usable. And then once we are sharing and reusing this data, we should have information about data provenance, that is, who created this data, where did it come from? And eventually, the most important things, we also have to comply with our funders and publishers, because they are the ones who give us money and they have requirements from us, which we need to follow. Now, what exactly entails data management and where is it coming from? So as you can see on my screen, there is a life cycle which the data goes through before the project is conceived. That is at the time when we are planning about what we're going to do in the project. And then it goes through various stages of the life cycle, whether it's collection of data, processing, analysis, preservation, sharing, and then finally reuse. So data has to be managed at all stages of the life cycle. And this has to be managed well. And data management does not apply just that you create a DMP, which is a requirement of the funders, and then forget about it. So no, you have to be managing your data well for it to be usable, for it to ensure that you are following the legal and ethical requirements, you're following the funding body requirements, that you're managing and storing and sharing in your data in such a way that it's useful to other people and not just you. Now, what is our ultimate goal in doing this? In doing this, we want to make data fair. And fair is an acronym which most of you will be familiar with. That is, we want to make data findable, we want to make it accessible, we want to make it interoperable, and we also want to make it reusable. Now, this fair is a set of principles which was published a few years ago. I think in 2016. I have put the DOI to the paper, you can access it. And it is a set of principles in which we are enabling our data to be open and transparent and reusable for the rest of the community. And to add to it, it is being increasingly adopted by our funding bodies. Now, there's also another concept that we would like to promote, that in addition to fair data, there is also the open data policies now, whereas we are moving towards open science. That is, we want our data to be available for everyone to access, use, and share. We understand that there are concerns about open data right now because people are not sure that they want to put their data out in the public before they have analyzed it, before they have published. But people usually confuse that if your data is fair, it's open. That's not true. Fair data does not mean open data. Data can be both, that is, it can be fair and open. It can be either fair, it can be open, or it can be none. So it is up to you. While we promote open data, we also want to ensure you that fair data is not open data. Now, what do we do towards this fair data management? How do we do that our data is fair and it is following the stages of the life cycle, and data is being managed at each stage of the cycle appropriately? So we divide it into three phases. For example, before the project, where you need to create a data management plan, which many of you might be writing because it is a requirement of the funding bodies or grants bodies, and you are costing data management. That is, how much of our cost should be assigned for data management. For example, do you have infrastructure in the university where you can save your data, where you can store your data, where backup is being taken. So you have to pay towards that. Or do you need to create your own system? So you need to cost all of that. Then during the project, you need to pay attention to organizing your data, storing and sharing your data, and then also security of your data. Some of you will be working with sensitive data, so you need to think about securing your data. And then after your project is completed, we need to think about data preservation, that how are you going to archive your data? Or now these days, even the publications ask you that your data should be available for at least 10 years after the publication date of the paper. So you have to be aware of that and take into consideration all this before you start your project. And this means these are all the steps towards fair data management. Ultimately, we need our data to be discoverable and reusable so that the community, the scientists can benefit from them, from the data, and then science can progress forwards. Now, during this each stage, if I go a little bit into detail about data management issues and best practices that you need to follow during these 3 stages that I said, that before the project, during the project, and after the project, I would say, as I said, before your project, you need to have a data management plan, which you have to do for a funding body, which you cannot escape. But this should be a document in progress. This should be a live document which you keep updating as the project goes on. This does not need to be written in stone or carved in stone. Once you're inside the project, what you need to be worried about or thinking about is documentation and metadata. We will talk about this in a little more detail in the next few slides, and I think Jasper will also touch upon this. Metadata is very, very important. That is something that is defining your data. Then you need to think about who are you going to give access to your data. This is happening, or who are you going to share your data with? This is going to happen during the project, and you need to have best practices and guidelines and rules set up how you're going to do this. You need to have policies. You need to have policies about how you're going to store your data during the project and how you're going to back this up, because it needs to be a safe system. And then, of course, after the project is completed, what is the plan and policy for long-time preservation and archiving? What happens after you have published the data? Who gets access to it? How are you saving it? So you need to have policy and best practices for all of these steps. And eventually, once you've published your data, you also need to think about dissemination and visibility. So do you have a URL? Do you have PIDs, digital object identifiers? So all of this has to be in your mind throughout the project, and all of this has to be taken care of at each stage. And to be honest, started at the beginning of the project. Now, I'm just going to spend a few minutes on metadata. Metadata is the most important thing. What is it? Is Jason Scott, he wrote in his blog, and I really love this. It's called a love note to the future. So it is something you're writing to yourself, to the person after you, or to the machine after you. So when somebody else comes to look into your data, this information about the data helps them understand what this data is about, so that we don't need the person who created the project or the data at the first place to be present there. So I really love this term, that metadata is a love note to the future, to yourself and to everybody else who will use your data. But what is metadata? In simple terms, it is data about data. That is data describing, if you have any numbers there, what are these numbers? What are its units? What is its realistic value? Then the context, that for what purpose was this data created? What was the experiment? What is its relationship with other data sets that have been created in this project or that have been used from some other project that is relevant to this experiment? Then data provenance is also very important, like who created the data? What was the data collection method? Where are these samples from? When were they collected? And then there is also the administrative metadata, which is who is responsible, when was it created, how can I and others use it? This basically involves a bit of policy making. So this, in short, you have to remember that metadata is key to your data being reused not only by the other people in the community, but by yourself in the future as well. I have just given a brief introduction to why this data management is important, but we have a very good tool that we have developed within Elixir, which is called the Research Data Management Toolkit. And it gives you a lot of best practices and guidelines to make your data fair. And there's also a domain page which documents microbial biotechnology, which is very relevant to the IBISBA project. So it follows the DBLT cycle, and it tells you how you can manage your data at each of these stages of the DBLT cycle. Now, what was data management for IBISBA? How did it become important for IBISBA? In our initial project grant document, or we had this figure where we talked about that we will have best practices for sharing our knowledge and assets. Whatever is created out of IBISBA, how will it be shared to the best possible, to our best possible ability? And then access to data. Who is going to have access to the data that is being created as part of the IBISBA, and how is that access going to happen? That is where IBISBAKHub comes in. So for IBISBAKHub, it's a single point of entry to the wealth of information such as protocols, models, workflows, or experiments that you have done. Then you have access, you have control over who you give access to your data or who you don't. And finally, you also have real-time project monitoring, that where am I in my project? What is the next step? So this is where FairDM-Seek comes in. I mentioned IBISBA, but we are starting with FairDM-Seek because FairDM-Seek is the parent to the IBISBAKHub, which I will come to in a minute. So what is FairDM-Seek? It is just an open source research data and metadata management catalog, and it has all these ways for you to store your data, save your experiment, get information about your experiment. You have yellow pages, which gives information about the people involved, the institutes involved. Experiments are organized in the ISA structure, where you can save and store your metadata about your samples. You have flexible sharing permission, so on and so forth. We'll get to that in a minute. And as I said, that these are some very key features of the FairDM-Seek. As you can see, that you can have information about the people involved. You can have data files of any type, samples, publications can be there, workflows can be there. So the whole point is that you have all the information about your project, about your experiment on this data management catalog or asset management catalog, which then becomes very easy for you to share with other people. So this is how we organize our information on the FairDM-Seek, which is we have this basic built-in structure where there is a program, you can assign projects, and then we have investigation, study, and assay. And all of these links that you see are the other information that you can associate with each of these levels. And then now coming back to IBISBAKHub, why did I say IBISBA Knowledge Hub at the beginning is the parent is FairDM-Seek, but you can use FairDM-Seek in various ways. You can build your own project hubs, and that is why we have built the IBISBA Knowledge Hub for the IBISBA and the bioindustry projects, because we want certain additional features, because we want to have it our own look and feel, and because we may need some features which are not required by other projects. So FairDM-Seek is a parent body, and from there we created our own hub, which is called the IBISBAKHub. Now, how does IBISBAKHub facilitate your data management? It does so by allowing you to organize your data or assets, which can be data files, protocols, samples. It helps you to store it for the future. It helps you share it among your collaborators. And most importantly, your data is secure and safe. So basically, IBISBA Knowledge Hub will allow you to make sure that your assets and your data is compliant with FAIR principles. Now, this is how the IBISBA Knowledge Hub looks. This is your front page before you log into this, and I've given the URL below. You can check that. This is how, when I talked about that, this is kind of an overview to give you a feel of how once you logged in and once you have your information there, how the IBISBAKHub will look. So I've taken a screenshot of the project. So you can see there are a list of projects here, one of which can be yours. And then if you just want to search for the people, you can see the number of people who are associated. And you'll also get information about these people, which projects are they associated with, which institutes are they associated with, what samples are they associated with. So you can get information like that. Now, how does IBISBAKHub, IBISBA Knowledge Hub, organize your experiment? We use the investigation study assay structure, which is a well-established nomenclature, and I've given the link to that here. You can check it out. So investigation is your basic experiment, or is your basic big project that you're going to investigate. Then the study, so your investigation will have multiple hypotheses, and these are divided into studies, and then each study will have its experiments associated to it, which is called assays. So there will be multiple outputs from each assay, from each study, and that's why we organize it into the well-established investigation study assay structure so that you can keep track of your data. And this ISA structure facilitates a rich metadata record for each step of the experiment. This is how the IBISBA hub, the ISA structure looks on the IBISBA hub. You can see the here highlighted in green is your investigation. The blue box shows you the study, and then the red one shows you assay. All these data files that are here they can either be the input or information about the input, or the outcome that has come out of this assay. So multiple assays are associated to a study, multiple studies can be associated to investigations, and then so on and so forth. For the sake of IBISBA, what we also introduced here is a new feature called the observation unit, which, for example, can be a plant in this case, or a microbe or a fungi, which was, let's say, infected. So from that infected microbe or fungi, you can take multiple samples. So that is so in addition to the investigation study, assay, and sample structure, we also have introduced the observation unit for you to put that information there. Then in IBISBA, we also allow you to record your samples. And for samples, we first need to build these sample types. Sample types are basically, we collect all the attributes that describe your sample. And then once you've created these attributes that describe your sample, then you can add samples to the Knowledge Hub and associate it to the project and associate it to the assay that they have come from. There are multiple ways of creating sample types. We will see this in the live demo as well. We can do it manually or uploading a sample template, or we can also do it by using the Fair Data Station, which Jasper is going to introduce today. And then to these sample types, you can assign your samples. Again, you can either do those manually, like this is a sample type here, the name of sample's glucose growth, and then you can add a sample there and information about the sample, that what temperature was it, what organism was it brought, extracted from, or you can also have spreadsheets which we can use to upload samples to the IBISBA Knowledge Hub. Especially this is very useful when you have a huge number of samples, so you don't want to be doing that manually. Now, these are some additional features that I'm going to talk about the IBISBA Knowledge Hub, is also that you can upload data files, you can upload standard operating protocols, you can upload documents. That is, there is no limitation of the format. Any format can be uploaded to the IBISBA Knowledge Hub. And if you have formats like Excel, Word, PDF, XML, or PNG, the regular formats that we have, if it's not a proprietary format, you can also explore and view these information on the IBISBA Knowledge Hub itself. A very important feature here is also some of the tracking versions, because sometimes we update. For example, I'm showing here is a kinetic model for incubation. So there are multiple versions of it, because we keep, sometimes when we're doing experiments and analysis, we keep changing parameters, or we keep changing the instruments. So we can have multiple versions. So we don't want to lose the whole trail of the other versions that we were using. So we can, so IBISBA Knowledge Hub allows you to keep track of all your versions that you have created. Now, the most important part, whenever I've given a talk about IBISBA Knowledge Hub, the concern has always been about confidentiality and access rights. What happens if I upload my data to the IBISBA Knowledge Hub and I'm not ready to make it public yet? I'm not ready to share it even with my other collaborators, let alone the world. So IBISBA Knowledge Hub provides you this flexibility. You can choose who to give access to your data. It's you who chooses who can view your data. You can keep it hidden, you can keep it open, or you can even choose to show some parts of it and keep the other parts hidden. So your data can be as open as you allow and as closed as you want. So as I said here, there is confidentiality at each level. That you want to show the project, just give general information, then you can choose that, okay, the public can view it. Or if you don't want, you can keep no access. But if you want somebody to see a certain experiment from your project or a certain file from your project, so you can give them access just to that particular investigation or study or assay. So rest assured, your data is in very safe hands when you upload it to the um, IBISBA Knowledge Hub. Again, I would like to reiterate, so you also have the option of sharing this data with your collaborators. Share with the person, you can share it with the project or institution, you can share it with the program, but this does not mean that your data is open. This is just making your data fair. Then eventually, why are we doing all of this? We want to disseminate and make our study, our science, our research citable. And for those, what we need is, for example, a URL, a digital object identifiers. They're called the digital object identifiers. And IBISBA Knowledge Hub also gives you the option to generate digital object identifiers for your data sets. And then those that digital object identifiers, you can share them with your collaborators. You might need them even when you are publishing your data with the journal.
5 more minutes, Munazah. You
Okay,
wanted
thank
to know,
you.
right?
Yes, I'm almost there. So what I wanted to reiterate and what I have not said from the beginning is that IBISBA Knowledge Hub is not a storage, it's not a database, it's a registry. So we store the metadata on the IBISBA Knowledge Hub, but the data can be stored elsewhere. So you can put the links to your data wherever it is. For example, there is a URL here, but only the metadata about that data that you have created will be on the IBISBA Hub. There is some flexibility to upload your data files, like I was showing Excel sheets or some standard operating protocols or your PowerPoint presentations associated. These can be stored on the IBISBA Knowledge Hub, but not huge amounts of data like the FASTQ files or the images that come out from different kinds of microscopic experiments. So we have to remember that IBISBA Knowledge Hub, it is a repository, it is a registry, it is an asset management platform, but it is not a database. So data can be stored remotely and then accessible from the IBISBA Knowledge Hub. Some of the other features which we also present is that we have integration with other softwares, like some of the SBML models that you might be aware of and might be regularly generating. These models can be simulated via the JWS Online and Copasi softwares that we have integrated them with the IBISBA Knowledge Hub, and so you can use them directly after uploading your model to the IBISBA Knowledge Hub. A couple of more features which I'm going to bring your attention to. So I talked about we have the investigation study assay structure. You can upload any kinds of data files of any format, whether it is the a doc file or an image file or a PowerPoint file. But we also allow for workflows, features like workflows. So this feature you can use to push out the data on the IBISBA Knowledge Hub to external computational platforms like Galaxy. Galaxy runs workflows for analysis. For example, if you're analyzing high throughput sequencing experiments, so you have workflows on Galaxy to do that. So you can push that out from IBISBA Knowledge Hub. Then we also have a feature sometimes some called collections. So we bundle some of the items together which are conceptually related, and then they can also be shared together as a collection. So as you see, all these features are there to make your data more and more fair, to make your experiment more and more fair. So it's all about compliance with fair data principles that we are trying to do with the IBISBA Knowledge Hub. We also have an API, so you may not want to use the publicly available platform like the IBISBA Knowledge Hub. You may want a local installation, and for that we have API, so you can have a local copy of the IBISBA Knowledge Hub or FedMSeq in your local environment. We also have the RDF representation now. I think Jasper will pick this up and talk about this more in his fair data when we are doing the fair data station, because these kind of formats are very important for integration with other platforms available out there. Now, in summary, when I started talk, I was talking about that these are the things that you need to take care of during the stages of the project. So IBISBA Knowledge Hub does help you. If you actively use the IBISBA Knowledge Hub, many of your data management issues will be taken care of because you're organizing your data via the ISA structure. You're putting the metadata about all the experiments and studies and essays that you're creating. You have multiple levels of access and confidentiality as and when you want. You have version control. You have the ability to share data, and you also have the ability to cite your data, create DOIs. So IBISBA Knowledge Hub basically helps you move towards verification of your data and take care of your data management issues. So I'm ending this section, and before we go on to the demo, I would like to know if there are any questions at this stage.
Any specific question yet? So then I would say you can continue with the demo.
Okay, so quickly, I'm just going to say what I'm going to show in this demo today is, so first of all, for getting started on the IBISBA Knowledge Hub, you need to register yourself on the hub, and it's a very simple process. As you can see, you just have to put in your name, email address, and register, and then your account will be approved by our administrators, and you should be on the IBISBA Hub. It's as easy as that. So I'm not going to do a demonstration of that. What we are going to do today is we're going to see how to create a project on the IBISBA Hub. Then we are going to populate that project with the ISA structure. And then we are also going to register. I'm going to do a demo of how you can register your data files, and then also samples here. But what I'm going to tell you is that if you get the hang of it, there's a lot of repetitive steps in there. You will get familiar just if you do one part of this demo with me, like create a project, then you can do populating a project in a much easier manner. So it's very intuitive and you learn as you go. And so today's demo will take place on this sandbox environment, which I'm going to open here. Can you still see my screen?
Yes.
Yes.
Okay, great. Thank you. So this is a sandbox environment. This is not the actual IBISBA Knowledge Hub. So this is also available to you. As you can see here, it says that this sandbox will be available till 10th of April. So you can go after the seminar, you can go and create your accounts and then start playing around without the fear of breaking anything, because this is a virtual environment that we have created just for the purpose of this workshop. So without wasting much time, what I'm going to do is, the first step as I said, is we need to log in. My account is already created, and I jump straight away. So the first thing, as I said, is that we are going to create a project on the IBISBA Hub. So on the PowerPoint slides will be shared with you. There is a link where all this workshop material is available, and you can then practice in your own time with all the material that is available there and create your own projects on this sandbox environment. So the first thing that we're going to do today is create a project. As you can see here, all this information, what you see on my screen right now, this is the first step that we are going to do. There are stepwise instructions available there, and you can do this in your own time. So we are going to do a project creation on the IBISBA Hub sandbox today. So the first thing, I will go here to the Create button and create a project here. And for the title of the project, I will copy it from here. The first thing we do is we copy the title of the project. We add some information about it, like the description that what this project is about, what are we going to do. It's like an abstract or a small introduction to these projects. And then we add an institution. If your institution is already registered, it will show up. If it is not registered, then you type it, and then you can add other information to it. For my case, I will add University of Manchester. And then because it is already registered, I don't need to put any other information. And then I hit the submit button here. And after this, I will go to the create button and the project will be created. Here we go. So the first part of this demo is done. So this is how easy it is to create a project. Now, once the project has been created, as you can see, there are multiple options here. You can go to the actions button, for example, if you want to edit something, if you want to add an organism which you have not specified here, I want to add project members. Firstly, I'll go here, I'll click on the manage project, and I'll see that if there's more information that I want to add. For example, if there are funding codes that I want to add, that let's say EU 2025 grant 123456, we can have that. If there's a public web page associated with your project, the project start date can be 7th March and the end date can be 2028. Let's see. We are associating with the program. So this project that I've selected is with this Aspergillus niger, so I can add this organism here. So usually you have to, if your organism is not shown in this pull down menu, you'll have to add it again by a very simple step of creating it. Then there are other options as I was telling you about, you can choose a license, that under which license, if you want your people to see your data, which license will you use? Then the most important thing here is that there is a default policy by which there is no access to public about anything about your data. At this stage, you can change it depending on how you want to do it. You can either keep it to view, that the public can view it, or you can give them multiple permissions. You also have the possibility of sharing it with a person. For example, I'm going to share it with Stuart here. And then again, you can choose what can Stuart see. Can he see a summary only? Can he get context? Does he have the right to edit and manage? So I can just say that view summary only, and I add Stuart here. And with that, I can update my project. And as you see now, we have here the Aspergillus niger. I added this information. And the next step that I would like to do is if I want to add some project members. Now, by project members, if your members of the project have already registered on the IBISBAKHub, we should see their names here. I'm going to add Jasper for the sake of simplicity, and then the institute with which Jasper is associated, and add him here and press the confirm changes button. And now you can see that Jasper has been added to the project. So there are two people now associated with the project. This is me, who has the administrative rights, and then this is who's the creator of the project, and then there is Jasper, who is also a project administrator. Do we have any questions at this point?
No, all questions have been answered already.
Okay, so just one additional information here. At any point, when you create a project, if you're not associated with a program, you might need approval. So usually what happens that when you create a project on the IBISBA Knowledge Hub, the administrators of the IBISBA Knowledge Hub, for example, me or Jasper or Stuart, we get an email that such and such person is requesting a project and we approve it. It's just an email approval, and then you will need to wait till we approve your project. So once the project has been created, you can see over here that your project has been created, and then we can move to the next steps of creating the ISA structure. As I said, we are going to create the investigation, study, and assay. So what do we do for that? So you can create an ISA structure. There are multiple ways of doing it. One way is doing it manually, the way I will show you. And then it can also be done through our other platform called the Fair Data Station, which Jasper is going to take you through. So let's go and start creating our ISA structure. I will again go on this create button and go on the investigation. And once your investigation is once you click on the investigation, a new page will reappear, and you will start filling this basic information about your investigation. So again, when you are playing with on the IBISBA Knowledge Hub sandbox, you can follow these stepwise instructions that we are providing you with, and I will copy this information again for naming the naming the investigation here. We will use the description that what this investigation is about, a very brief description. Then we will, these are the important steps. Now, this investigation belongs to a project. What project does it belongs to? An inventory of Aspergillus niger, which I just created with you. So once you do this, you will get this banner which says that this investigation has a default sharing policy, which I just showed you a minute before. Do you want to apply it? Okay. And you can always change it later to your whatever your priorities are. Then you can put in the investigation position. Sometimes when you are doing multiple investigations, and when you see the view, you want some investigations to be the first one, you would want some to be the second one. So you will write the investigation position. I'm going to put it as 1 right now. I'm going to change the permissions that the public can view this, and Stuart maybe can edit it. You can add more creators here, and then you can also add tags which can be like microbial biotechnology, just for you to identify what, or somebody else who sees it later, to identify what is this project about, or what is this investigation about. You can also add discussion channels, maybe URLs, for example, if you're having a discussion about this on some Slack channel, but this may not be that important to you at this point. And once I've put in this basic information, I'll hit the create button and my investigation will be created. But before we say, okay, it lets you know that you have allowed Stuart to view and edit the summary of the project. Are you okay with it? This is just to address the confidentiality issues that people have. And we create an investigation. So voila, that's our investigation there. And next, as we know, because we are going to follow the investigation, study, and essay structure, now we are going to add a study to this investigation. Now you can do a study by 2 ways. Either you go back to the create button and hit study, or what we can do here is we can add a new study to this investigation straight away from this page. And that's what I'm going to do. I'm going to add a new study here. And again, it will tell us about that there is a default sharing policy. Do you want to apply it? Okay. Now going to add study. As you can see, these are very repetitive steps. If you get a hang of it the first time in creating a project, then it will be very easy for you to create the rest of it. So let's see here. I'll copy a title of the study that I have put in here. This is in silico investigation, and I will put this here. And then we are going to add some other information that is there. So sometimes people do have extended metadata, which is like the MyRP metadata format, which is already available for the plant community. So maybe if IBISBA or the biotechnology community has such a metadata, so we allow that it's already inbuilt into the Fed in our IBISBA Knowledge Hub, and then you can get a selection from here. So it is already associated to an investigation because we started it from that page. And now, again, I can put a study position of 1. And again, we come to sharing permission. What do I want? What kind of permission do I want to give other people? Maybe I can let Stuart manage this study as well, because maybe he's the one who's going to add some study material to it. Like before, you have other options to add here, and then you hit the create button. Again, it will tell you that asking you that this is the sharing permission that you have chosen. Are you okay with it? And the study was successfully created here. I will go back to my projects. Yeah, so you see now we have a project here, we have an investigation, and we have a study associated to it. So now the next step would be to start to add an assay to this, which is the actual experiments that will be carried out to prove your hypothesis that you have stated in the study. So again, you have the option of doing it directly from here, from the Create button, or you can add a new assay to this study. At the study level, you can also add an observation unit, like I mentioned, if you have one observation unit to add, or you can directly add a new assay. What I'm going to do is I'm going to add an assay. So when you hit the Create Assay button, you will get two options. One, it can be an experimental assay, or it can be a modeling assay. So this helps you because this is kind of our way of categorizing into what kind of experiment was carried out. So for now, let's go and choose the experimental assay. As usual, we have to fill in the basic information about the assay, which I've already put in this document here, which you have access to. It is already associated to the study in silico prediction. We'll give it an assay position. This was an experimental assay, and you can choose further what kind of assay it was, and you can say what kind of technology type was used. Some of your technology types may not be here, but you can add them. And then we add the organism to which it is associated. Select the sharing permissions. We are just going to keep it like this. And the rest, as you can see, now to the assays, you can add many more types of information, such as you can add data files to it, which we are going to do. You can add samples to it, which we're going to do in a minute. You can add standard operating protocols to it, so on and so forth. Or you can upload them separately and then just assign it or associate them to the assays. And here we hit the Create button, and we can go now and create our assay. So as you can see, now our investigation has a study and it has an assay. And if we go back to the related items here, it will keep showing to you that there is a project, there is one investigation associated with it, and there is one study associated with it. And in this button overview helps you to view your project in the tree structure that we are going to do here, that we are seeing here. And after this, what I'm going to do is now I'm going to show you how you can upload data files and associate them to the assays that you have created. So what I'm going to do here is I'm going to go and choose a data file, which is already in the workshop material. I'm going to create a data file, which this data file can be anything. It can be an Excel sheet, which is the output of your experiment. It can be raw numbers that you have, or your final analysis that you have done from the raw numbers that the experiment has shown you. So we'll go and create choose a new data file. So your data file can be on your local computer. Okay. So somebody has already tried it, that's good. So I'm going to choose a data file here. For example, you can see there are multiple data files here. For example, they are named D1, D2, and D3. So I'm going to choose mass spec results here, upload it, and register this data file. And now, like before, we have to add this information about what this data file is about. So if you go to the section 3 of this document, which is available to you, there is information available about what this data file is about. So you can take this. You can put some description about it. And then you can now assign it to which project it belongs to. Of course, we were doing an inventory of Aspergillus niger secretome, and we are going to add it here. And then we can go to the next, and you can type annotations. For example, it's data. You can data format. Let's say it's an Excel file, and we add that. And then again, you have the right to choose licenses, what kind of sharing permissions you want to do. Do you want to add more creators? And then you hit next. Again, it will ask us if you want to associate it with any essays that you want to do. We are going to associate it with and this is other related information, like you want to put any publications that are associated with it, the observation units, if you want to associate with it, there's already an observation unit available here, we can add it. So there are now data files. This data file is, just a minute, scroll, yeah, that should be done. And we can go back to our project now. So this is the data file that we have uploaded and it is available here and it is associated to our investigation. So this is our essay here. We can also add a new data file directly from here. I'm just repeating the same process via a different route. You don't need to go to the create button every time. You can just do it from where it was, from where you were. And you can see this now, this mass spec data file that I just uploaded is now associated to the assay signal peptide predictions. So this way, your ISA structure has been created. You have the investigation, you have the study, you have the assay, and then you have the data, so data files or data output files. You can also upload PowerPoints or other, or even minutes documents that you have to this step. Now, after we have done this, are we doing all right on time? Do we have any other questions?
No questions except the ones answered. There is one left, but that's a bit more general one. I will come back to that later. And in terms of time, there is 5 to 10 minutes for you to go.
Okay, so now what we're going to do here is, this is the last step of my demo here. One thing that I will see that you might have, it might seem very overwhelming right now, but you might have noticed that the steps are repetitive, that once you do one part of it, you can then get a hang of it. So it's very similar, it's very intuitive, and you can keep adding information as and when you like. Now, in the third section, in the last part, what I'm going to do is I'm going to try to demonstrate here how we are going to register samples. The samples that we are going to associate with our experiments. As I said in my presentation, that to register samples, we first need to create something called sample types. Now, these sample types is information about your sample that you're going to create. I'm going to show you here in the like what does a sample, so now there are multiple ways of uploading sample types to our experiment here. One, it can be through it can be manually defining the attributes using a form, which I'll show in a minute, or it can be done by uploading a template, which we have sample type template, and you can use that. Or we can also do the same step by Fair Data Station. Now, this spreadsheet and the Fair Data Station are very, very useful when you have a batch upload, like when you have hundreds of samples and you don't want to do them by hand. So you can do it manually when you just have a few samples, but the rest of it, to automate the process, there are other ways of doing it. Now, what we are going to do is we are going to create some sample types for our samples today. And we will go to the create button again, and we will create sample type. And from here, what we will use is, so you can either do it manually, as I said, that is by filling this form here. You can do the attribute, write attributes, write the type it is, and then description of that sample type. But what we are going to do, we're not going to do it manually. We're going to use a template. So for that, we will go to the use spreadsheet template tab, and we are going to put some information about that, about the sample type, that what it is going to be about. Okay, I have kept some here, information here, which we are going to. Description, we'll assign it to our project, that is inventory. Okay, we'll see what the sharing permissions is. We'll just keep it that people can just view it. And then we choose this template, which we have already created, which is here, D3. And that's it. We are going to create our sample types. At this stage, you can see all this information. Let me also show you what was inside this template information. So this is the sample name, strain, label, substrate, and type. These are all the sample type. These are all the attributes that are defining your sample. So when we are creating the sample type, we are taking all this information and putting it into our IBISBA Knowledge Hub. So this is what you will see here now. When I uploaded this file, it has extracted this information, sample name, strain, label, substrate, type. And at this stage, you can either choose which attributes you want to keep, which you want to delete, or if you want to add something here, or if you want to add some more information, you can do that. And then we can update our sample type. So hence our sample type here is created. Now, the next thing obviously that you would like to do is add samples to the sample type. Now you can do that in two ways. Either you can go and create sample from here, just create sample and then add sample names and add the information according to the sample type we have created. But that's not what we are going to do today, because we don't expect that you will have a few samples. We always expect that the possibility is there will be more samples. So instead of doing this, what we're going to do is that we are going to upload the samples via a template. This is the template that we have created, and that we are going to upload this to our IBISBA Knowledge Hub. So we again go to the create, and now this time we will choose create samples. And for this, we will have to select a sample type. And as you know, we just created this demo sample type, and we are going to choose the sample type here. So this is one way of doing it. The other way is we directly go to the data file, and in this data file, we choose our sample data file here, upload it, and then we go through the similar process of choosing information and adding this information, which we are going to do here. Assign it to the project. Data format. And then you can hit the Extract Samples button here. Choose the sample type, and the samples are being extracted. This is just for you to view if everything is for reviewing your extracted samples. This is what we had in the file, and now they're available in here. And this can be linked to assays here. And once we see that everything is okay, we don't want to do anything here, we don't want to add or cancel anything here, and then we can confirm. It is still creating these extracted samples. And we can view created samples. This is how it is done. Now, what is also a possibility here that you can also explore your samples here once you have uploaded them. So now you can clearly see here that what your sample type is and what the information that you had in the spreadsheet is here about your samples. Now, if we go back and see how it should look, I will show a fuller example so that you get an idea of how it should look in the end, which is this is how your structure will look like. Once you have followed these steps, you'll have I don't know if you're able to see my full screen, but I'm just going to scroll through. So you will have your investigation, you'll have your study, you'll have your assays, and all of these and all the assays will have these outputs related to it. For example, there are 16 samples now associated with signal peptide interactions, and there are all these Excel sheets that you see associated with them. These are the outputs of this experiment. Then your next study here is culture experiments. It has its own assay, liquid chromatography associated to it, and the outcome of these and samples associated with it. So this is how your tree-like structure will look like, and this is what the ISA structure is. So these are the three things that we wanted to do. I wanted to show you today, that is how to create a project on the IBISBA Knowledge Hub, how to populate it and create an ISA structure, how to register your data files, and how to register your samples. I understand this is overwhelming. There are multiple ways of doing it, but that is the reason we are providing you the sandbox where you can play around. I'm just going to, so where you can keep playing around till the 10th of April and at your own speed, and you will have access to this workshop material, which is a stepwise instruction of creating the project, populating the project, and registering data files and samples. So you can do it at your own pace, as we recognize that this is a lot of information that we're giving out to you.